Back to chapter 23, folks. So we can do a few videos looking at protein kinase associated receptors, and much of what we talk about will be receptor tyrosine kinases, but we'll probably see some serine threonine kinases as we get going. So protein kinase associated receptors, they're not only receptors, but they have their own inherent biochemical activity in as much as they function as kinases. Now remember, kinases can transfer phosphates to substrates, usually proteins, but in some cases lipids, but we will be focusing on protein kinases. So when a ligand binds to the receptor, the kinase activity is stimulated, so the kinase activity of the receptor is turned on. And so once the kinase activity is turned on, that then results in a signal transduction cascade into the cell, and the cell has a response. Now, like I mentioned, kinases are either tyrosine kinases, in which case they phosphorylate the hydroxyl residue on the R group of a tyrosine amino acid, or on the hydroxyls of serine and threonine, in which case they're serine and threonine kinases. So tyrosine kinases are monospecific, the tyrosine, and serine threonine kinases are called dual specificity kinases. So what are these kinases doing? Well, for cells to divide, they need enough nutrients, so you know, all the metabolites they need for growth, and they also need signals to stimulate their growth as well. So if you have cultured cells in vitro, so just growing in a dish, you can give them all the nutrients in the world, and they won't grow unless you provide them with blood serum. And so in that blood serum are messengers that stimulate cells via cell surface receptors, a lot of receptor tyrosine kinases and threonine serine kinases. And so these receptors are responding to growth factors, these messengers in the serum. So what exactly is serum? So let's kind of start off with plasma to start with. So if you take whole blood, looking over on the left, if you take whole blood and you centrifuge it and you pellet down the erythrocytes, and then you end up with a thin layer of platelets and other kind of white blood cells, then the straw-colored fluid that's left is the blood plasma. And so that includes all of the clotting factors that you find in blood. Now that plasma will not support cell growth in vitro, and so that's not adequate for growing cells in vitro. So that's plasma, because what we need is serum. So serum is what's left over if you take whole blood and you then clot it. And so the serum is basically the plasma lacking all of the clotting factors, primarily fibrinogen or fibrin. And so that's the serum there. So in that serum, there are a lot of these growth factors. One of the factors we'll look at is a growth factor that's secreted by the platelets that stimulates fibroblasts to form new connective tissues. And so that's called platelet-derived growth factor. And so there is a ton of platelet-derived growth factor in the serum that can be isolated from whole blood. And so the receptor for platelet-derived growth factor, or PDGF, is a receptor tyrosine kinase. So we'll probably take a look at receptor tyrosine kinases in the form of the PDGF receptor in a little bit. So just to give you an idea of some of these growth factors that you find in serum and the things they do, epidermal growth factor acting on a whole bunch of different types of cells, and its receptor is a tyrosine kinase. So I'll let you just look through the list. You don't have to memorize the list, but what I'd like you to take away is the range of cell types which respond to different types of growth factor, and note that there are a large number of tyrosine kinase receptors that mediate the signaling via these growth factors. So let's take a real quick look at a prototypical receptor tyrosine kinase. Like we say, many of the receptor tyrosine kinases trigger this chain of events in the cell that ultimately culminates in things like cell growth, cell division, and in some cases differentiation, depending on the nature of the receptor that we're looking at. So receptor tyrosine kinases are often a single polypeptide with just one transmembrane segment. So here is the whole receptor. You can see there's an external domain, there's the transmembrane domain, and then there's a tyrosine kinase domain, and then there's this large carboxy terminus cytosolic tail. And so the extracellular part of the protein is where the ligand binds. So the ligand will bind up here. And then on the cytosolic side is the tyrosine kinase. And you can see there's this whole run of tyrosine amino acids in the cytosolic tail, and those will become important in a moment. In some cases, the receptor and the tyrosine kinase are separate proteins, and then they associate upon activation. But here we're looking at a receptor tyrosine kinase, which has got a built-in tyrosine kinase domain. So ligands bind to the external ligand binding site. So here we're looking at the epidermal growth factor receptor, and so epidermal growth factor will bind in this ligand binding site, and you can see in the three-dimensional model over here, here is one EGF receptor in purple, and here's another one in pink. And so in many cases is what happens is that the binding of the ligand causes the individual proteins to dimerize, so they come together. So once you get ligand binding, two receptors come together, and then the tyrosine kinase domains phosphorylate one another, and this is called autophosphorylation because these receptors are the same. They're the same protein, so it's this protein phosphorylating this one, and vice versa. And so you end up with all these phosphorylated cytosolic tails, and that's critical for initiating the signal transduction cascade, which will result in the cell response. So what we've looked at here really is just a little bit about the nature of the growth factors and their abundance in blood serum, and then the overall structure of the receptor tyrosine kinase. So as we come back and look at some more of this, we'll start getting into some more details. So I'll see you for that soon.